Well, thank you very much for the introduction. I'm going to move forward. I have to read this. OK. So I have to present it at least. So I'm going to speak about a dual platform. A dual platform of, on one side, the use of T cells as an active reagent to kill cancer cells with a specific head called a chimeric antigen receptor to target and make all the recognition of the tumor. And on the other side, I'm going to talk about gene editing and most specifically talin nucleases, which is TAL effector nucleases. So what are CAR-Ts? CAR-Ts, it's a new way to try to treat cancer through immunotherapies using T cells. So the T cell is an extremely powerful tool to kill other cells. A T cell can kill in a row approximately 10 to 15,000 cells, depending on fitness. We don't know exactly what's the maximum fitness of a cell. It depends on the individuals and also the exhaustion of the cell. But it has a second effect also, start dividing, but 10, 15, 100, 150,000 fold, also depending on fitness. So when you insert inside this cell a chimeric antigen receptor that has a tip at the top of it, called like a SCFV that can recognize very specifically tumor-associated antigen, it will trigger this killing effect, and it will start destroying all the cancer cells that have this target at the surface. On the other side, we're going to use another technology, which is like TAL effector nucleases, to try to remove certain genes or replace certain genes inside a T cell to make it more effective. And the fact that the ability of using gene editing to modify these T cells and make it more and more powerful is to do a series of different things. For example, suppress the alloreactivity of the cell to make them off the shelf. So you can take T cells from a healthy donor and give these T cells to a receptor, like a receiver that don't have enough T cells, for example. But you can make the cell also resistant to certain drugs. So you can remove receptors that can recognize these drugs, such as CD52, to make them resistant to alemtuzumab, deoxycytidine kinase to make them resistant to PNA, such as fludarabine. Or you can suppress also cross-T cell reaction, checkpoint inhibition, a lot of different things. I'm going to show you some of these features. So the way we engineer CAR-T cells, like off-the-shelf CAR-T cell, we get started with healthy donor apheresis. So you take white blood cells from healthy donors, you extract the T cell, and after activating the cell, currently in GMP manufacturing, the product we're working on and that are currently in the clinic, we insert the CAR randomly. But the next generation will not be random. With a lentiviral vector that insert the CAR inside, then after this, we use an electroporation system to introduce the gene editing technology inside the T cell that will remove specific genes, for example, the T cell receptor, and then we amplify the cell, filter the cell to remove all the TCR positive cells, and at the end you have a series of vials that can be immediately given to the patient. It's not a personalized medicine, it's what's called universal CAR T cells, so UCART. We've already manufactured a series of these CART, a CD19 one, CD123, CD22, in a full QC system. So the pipeline of product is the following. Currently, there are two products that are in clinic in phase 1, UCART19. I'm going to show you some data on UCART123. The other that will enter into clinic very soon is called UCART22, and a series of other drugs that will go in the clinic in coming year, called, for example, UCART-CS1, in a series of different indications ranging from acute myeloid leukemia to acute lymphoblastic leukemia, to multiple myeloma and other non-Hodgkin lymphoma type of tumor. So let's talk about, for example, the proof of concept of these like gene-edited CAR-T cell. We have this current clinical trial that is ongoing. The first patient with this pediatric ALL was dosed in June 2015. It's a compassionate use. This patient is still alive today. And there is 2 trials that are ongoing currently in the United States and Europe. And we're continuing to recruit. There is like 7 pediatric patients that have been presented and 7 adult patients. I'm going to show you some of the data of the pediatric patient, which are extremely interesting. Most of these patients are last line of treatment, means that like, they failed most of the current standard of care, first line treatment, second line treatment, fail like, for example, bispecific antibodies or even bone marrow transplant. 100% of these pediatric patients fail a bone marrow transplant and they have been treated afterward with super aggressive type of ALL. After the infusion of UCART19, we have 100% of complete remission at day 28, with complete remission. And then the adverse effects, which are like over grade 3 cytokine release syndrome, is fairly mild. We have only 13% of this with no neurotoxicity. You do have a relapse rate afterwards. It means at 6 months follow-up, there is a 30% relapse rate, which either CD19 positive cells or CD19 negative, so one CD19 negative. The CD19 negative, it means that the CAR T cannot act on the cell because like the target is lost. So we have like other data on pediatric patients, but I'm going to more focus on this presentation on the gene editing of T-cell next generation product. So here is like UCAR123, the next product we're developing. I'm not going to speak all about them, but this one is targeting essentially acute myeloid leukemia. We have been starting dosing patients in MD Anderson and also Cornell University, Cornell Medical Center in June and July last year. We had one death, unfortunately, with BPDCN because one of the patients had a grade 4 CRS and passed. So like the expansion of the CAR was so strong that the patient passed. We had a hold from the USDA, then the hold was lifted. And then following the lift of the hold, we restarted dosing patients and we're re-escalating this. It's a very interesting drug because it's extremely powerful against CAR T, against the AML cells. So you see that the standard of care, it's the blue line, what's called aracitidine or C-terabin. When you infuse, for example, UCAR123 or UCAR at 2.5 million cells or 1 million cell, you see that all the cancer cells, all AML cells are totally obliterated. And one of the lines we have to beat in the clinic is the blue line here of aracitidine that gives you an expansion in overall survival. When you infuse UCAR123 in there, you have an overall survival that is almost doubled or tripled compared to the standard of care, which gives also a lot of hopes. One of the real concern also is that like CD123, the target of this CAR, is expressed on the surface of myeloid progenitors. And the real question, is it going to harm the normal bone marrow stem cells? So the thing that we decided to do is like to do a mixed model where we inject UCAR123 or control cells together in mice that have either primary AML cells and normal bone marrow, human bone marrow cells. And you see that in peripheral blood, in the mice that has been treated with UCAR123, all AML cells are gone while normal cells continue to grow, which is not the case in control mice. And interestingly, when you check, for example, the bone marrow of these mice, the human percentage of human normal cells increase in the normal, like when they're treated with UCAR123, while all AML cells are gone. In the control one, at 36 days after treatment, this is not the case, and you see that the bone marrow is like diminished and AML cells start growing. So we're currently in clinical trial with these patients, and we're expanding in different type of protocol, pediatric and adult use of UCAR123 in AML, and there is a very strong medical need. So CS1 also is a new CAR. It's interesting because it's not a CAR that you can do without gene editing, for the simple reason that the T cell on the surface contains CS1 in there. So if you put a CAR that recognizes the target that is on the T cell themselves, they will start self-destroying themselves. And that would induce what's called a cross T cell reaction. So the thing that we have to do first is remove from the T cell by gene editing the target. So CS1 is removed. And you see that when we do the knockout of CS1 here, you see that the CAR, the cells that are transduced with the CAR and the normal non-transduced cell grow approximately the same. You don't have 100% knockout efficiency. While if you do this without gene editing the cells, the CAR self-destroy themselves and don't grow the same. But you see that the shape of the cell and the spectrum of these cells don't change that much compared to normal cells in general, so untransduced cells. So we know that the phenotype is unchanged. When you infuse these cells in mice, you see that if you put the cells, the multiple myeloma cells that have been treated or untreated, or plasma cell in leukemia, you see that just after the injection of CS1, you see that the total disappearance, and this is magnification of this phase here, total disappearance of multiple myeloma cells. However, in control, you see that the multiple myeloma cells grow again, while in treated with UCAR-CS1 cell, the multiple myeloma cell don't grow again, and we monitor the M protein all around the growth of these cells. The interesting thing with an off-the-shelf CAR T is that it's a normal drug. It's not a personalized drug, and so you can inject it as much as you want. If you make only a single injection of the CAR, you see that you have a better improvement of the life of the mice compared to control. But if you do a double injection, then you see that the life is expanded. Either if you do a dose at 3 million cells or 10 million cells, you see at 10 million cells, the mice almost don't die. So it's very, very encouraging results showing multiple and repeat dosing of these type of product. This product is supposed to enter clinic in less than a year from now. So let's talk a bit about the TALEN gene editing technology and what is the future of these CAR T cells. And the ability to engineer cells, for example, removing gene is the first step, but the potential to re-engineer these cells in a very sophisticated way can be really spectacular. And you see here that we can engineer or target a lot of different genes, always at a very consistent level of knockout. And most of the time, the gene, the off-site targeting is close to background, and there is no off-site targeting, so it's extremely specific, with over 60% double knockout, or even going to 90% or 95% double knockouts. Why the ability of using, for example, a protein-DNA interaction is interesting? Because protein can be engineered and re-engineered. So we have a collection of over 400 RVDs. RVDs are these variable D residues. Each of them recognize a specific base pair on each side of the tail end. And you can, for example, take one of these RVD and changing by another RVD that is called non-conventional RVDs. In order to suppress, for example, here, if you do a knockout of PD-1 together with the TCR alpha, so the track target, the TCR alpha, sometimes you can have the left arm of the PD-1 tail end that can cross-react with the left arm of the track tail end and cut an off-site, on non-owned off-site. And the ability to re-engineer these RVDs and re-engineer the protein allow you to suppress totally, so we can replace certain of these RVDs to totally suppress the ability of one of these RVDs to recognize the base pair that cross-react with the off-site. Here you see that there are like series of different type of off-site. This is PD-1, this is track, and these are the two off-sites we're talking about that potentially can be cleaved. They're cleaved by the normal PD-1 also, but also cleaved by the potential two track and PD-1 combination. And by changing initially the different type of tail end that we engineer, we can totally suppress the off-site targeting by these tail end, by re-engineering the protein itself. So it's a very flexible potential to recognize very specifically DNA. Not only this, but also the engineering of the tail end and the ability to cleave very high target is something that is important. Here you see that we have close to 95.2% of DNA of cleavage inside a T cell. If you have over 95% of signal knockout, what you need to have is a high recovery of the cell. So post transfection, you see that the mock cell survive at 92.3%, while track talent survive, like cells that have been treated by the tail end, survive at 90%. if you fry too much the cell during the electroporation or gene editing procedure, it will be very tough to the cell to recover afterwards. So here I'm going to speak about what the future of these type of technologies can bring. The idea here is, for example, to insert the CAR at the TCR locus. So that's the gene targeting of the CAR. It's not the random insertion I was talking about by lentiviral vector. But on the other side, what we're going to do, we're going to knock in the IL-2 gene inside the CD52 gene inside the cell. And what's going to happen, on one side you will have the CAR that will be expressed, that will induce an anti-tumor effect, but also that will induce a reaction against the CD52 promoter and will induce the expression of IL-2. You see, when we challenge the tumor, like the mice with like the CAR T with a tumor, or T cell with a tumor, you see that the CD52 expression is enhanced. And if you replace the CD52 expression with IL-2, you see the IL-2 is also expressed the same way. And IL-2 have a very potent and powerful anti-tumor effect. The problem is that if you give IL-2 systemically, you will have very high adverse effect. In this case, you have a local on-target IL-2 secretion. And you see that the anti-tumor and the anti-leukemic activity of an IL-2 charged or armed CAR T cell is extremely powerful. You can also re-engineer the cell in order to make it not visible, for example, to NK cells. So on one side, you will insert the CAR at the TRAC locus. On the other side, you will insert an NK inhibitor at the beta-2 microglobulin endogenous locus. Therefore, the allo cell cannot be recognized by the NK cell normally because like you have the MHC minus, the NK cell can attack the cell. But if you put an inhibitor, the allo T cell cannot be recognized as a non-self, but the NK cell, because of the lack of MHC, and the NK cell cannot recognize this because you can add also an inhibitor. So we can ask ourselves how powerful, how much this type of gene editing can happen in cells. And this requires the double knockouts. So you can see here that the TCR locus TALEN can knock out at more than 96% the TRAC locus. But when you insert, for example, beta-2 microglobulin at the TRAC locus with a TALEN, this happens at 99.9%. And the TRAC plus beta-2 microglobulin, you can have close to 88% of double knockout and 68% of targeted integration at each locus simultaneously. That's probably the first steps of what can be imagined as a kind of a synthetic biology inside a T cell in order to re-engineer the cell to make it not visible to the immune system, more powerful, secreting certain specific molecule at the challenge of the T cell, etc. And it marks also like the first step of a new era in the way we can reprogram the CAR T cell to make them real cancer-killing machines. Here in this case, you see, you can see that we have the TCR locus that has been targeted, the CAR inserted at the TRAC locus, the NK inhibitor, and all of this has been done at over 60% double knockout and with the 80 and 72 targeted integration at each locus. We're not talking about just knocking out the T cell, but just replacing a gene by another one inside the T cell. And that's probably one of the start of what this kind of technology is going to be in the future. So maybe like some conclusions. So the TALEN gene editing allows like GMP production of allogenic T cells with initial efficacies. You see that is on par with autologous CAR T cell. We have 100% complete remission for CD19 CARs. The TALEN display a very high level of specificity with 19 with 1, 2, 3. The first patient dosed is still in complete remission today. The second one that was dosed in November 2015 is still in complete remission today. So these are very spectacular results. In a single shot, you can give like complete remission. But the specificity of given TALEN and the potential offsite, because it's a DNA-protein interaction and not an RNA-DNA interaction, such you can have, for example, for CRISPR that we do use for the past 5 years, you can suppress all the genotoxicity risk in re-engineering the protein itself, and that's the power of the protein-DNA interaction. And finally, you have a very high level of multiple knockout and knock-in that open a vast array of possibility to improve clinical benefit of the CAR T cell. What I showed you here, and probably the first CAR T that when we filed an IND, I remember when we filed the IND in front of the EMA, the European Medicine Agency, they said, wow, it's like the most sophisticated drugs that we ever see. So you add a gene, you suppress 2 genes, and like this is complex. This is probably the prehistory of what the medicine is going to become with gene editing and the use of cells as the next generation medicine against cancer. And this will expand probably a pipeline against, this is only liquid tumors, but probably solid tumors will become a real power in this space. And solid tumor can probably be cured by not a single injection, but repeat injection with these type of drugs. Thank you very much.